Viruses are the most abundant biological entities on Earth. In other words, nothing else that resembles life exists in greater numbers. Not animals, not plants, not bacteria. They are everywhere, in the oceans, the soil, the air, and even inside living organisms. Scientists estimate that every teaspoon of seawater contains millions of viruses. That means if you scooped a small spoonful of ocean water, it would hold more viruses than there are people in many large cities. Because they are incredibly tiny and invisible without powerful microscopes, most people never think about them unless they cause disease. But in reality, the vast majority of viruses don't infect humans at all. Most infect microbes and bacteria. So while viruses often get a bad reputation, they are actually a constant and natural part of the planet's ecosystem, present in numbers that are almost impossible to imagine. Scientists estimate that there may be around 10 to the power of 31 viruses on Earth. That number is so large that it's hard for the human brain to truly grasp it. Written out in full, it would be a 1 followed by 31 zeros. To give a sense of scale, that's more viruses than all the stars in the observable universe, which scientists estimate to be around 10 to the power of 22 to 10 to the power of 24. In other words, there are likely millions of times more viruses on Earth than stars in space. Most of these viruses exist in the oceans, where they infect microscopic organisms such as bacteria and plankton. They quietly play a huge role in controlling populations of these tiny life forms. Many people think viruses mainly exist as disease-causing germs, but human viruses are actually just a tiny fraction of all viruses on the planet. The overwhelming majority live unnoticed in nature. If all the viruses on Earth were lined up end to end, they would stretch across the universe. This sounds unbelievable, but it comes from the enormous number of viruses that exist on our planet. Remember that scientists estimate around 10 viruses on Earth. Even though each virus is extremely small, many are only about 100 nanometers wide, which is thousands of times thinner than a human hair. Their combined length becomes astonishing when multiplied by that huge number. When researchers calculated this, they found that lining them up could stretch for about 100 million light years. A single light year is the distance light travels in a year, about 9.5 trillion kilometers. To picture it another way, the chain of viruses could travel far beyond our Milky Way galaxy and into distant clusters of galaxies. It's a powerful reminder of how incredibly numerous these tiny particles are. Viruses occupy a strange gray area between living and non-living things. Many scientists do not consider viruses fully alive, even though they contain genetic material and can evolve over time. The main reason is that viruses cannot function on their own. Living cells, like those in animals, plants, or bacteria, can grow, produce energy, and reproduce independently. Viruses cannot do any of those things without help. Outside a host cell, a virus is basically an inactive particle, almost like a tiny biological package. Imagine a USB drive filled with instructions but unable to run them without a computer. That's similar to how a virus behaves. It carries genetic instructions but needs a living cell to execute them. Because of this unusual behavior, scientists still debate how viruses should be classified. They share some traits with life, but they lack many essential features that define living organisms. Unlike living cells, viruses cannot reproduce by themselves. They must invade a living host cell and take control of it in order to make copies. Think of it like a factory takeover. A virus enters a cell and injects its genetic instructions. The cell's machinery, normally used to make the cell's own proteins, gets hijacked and forced to build new virus parts instead. The cell unknowingly becomes a virus-producing factory. Without a host, this process simply cannot happen. A virus sitting on a surface or floating in the air is basically inactive, almost like a seed waiting for the right conditions. This is one reason scientists debate whether viruses are truly alive. Living organisms can reproduce independently, but viruses completely depend on other cells to multiply and spread. Once inside a cell, however, a single virus can quickly lead to thousands of new virus particles. A virus is surprisingly simple. At its core, it mainly consists of genetic material and a protective protein shell. The genetic material can be either DNA or RNA, which contains the instructions needed to make more viruses. You can think of a virus like a tiny instruction package. The genetic material holds the blueprint, while the protein shell protects those instructions as the virus travels between cells. Compared to living cells, viruses are incredibly minimal. Typical human cell contains thousands of different molecules and complex structures, but a virus may contain only a few basic components. Some viruses have only a handful of genes, while humans have about 20,000 genes. This simplicity is one reason viruses are so small. Many are thousands of times smaller than a typical cell, yet their tiny genetic instructions can completely take over a cell's machinery once infection begins. As we move forward, feel free to share where you're listening from, or leave a quick thought below. And if this channel means something to you, subscribing is a small way to support the journey. Some viruses have an extra outer layer called a lipid envelope. This envelope is made of fatty molecules, similar to the membrane that surrounds our own cells. What's surprising is that viruses often steal this layer from the host cell they infect. When new virus particles leave a cell, they wrap themselves in a small piece of the cell's membrane, creating this protective coat. You can imagine it like a thief wearing a disguise made from the victim's clothing. Because the envelope comes from the host cell, it can sometimes help the virus blend in and avoid detection by the immune system. However, this envelope also has a weakness. Since it is made of fragile fatty material, it can be easily damaged by soap, alcohol, or disinfectants. That's why washing hands with soap is so effective at destroying many enveloped viruses. Viruses are not limited to infecting humans or animals. They can infect almost every kind of living organism on Earth, including animals, plants, fungi, and even bacteria. Many people assume viruses mainly cause human illnesses like the flu or colds, but that's only a tiny portion of the viral world. In reality, most viruses actually infect microorganisms. For example, many viruses specialize in infecting bacteria living in oceans, soil, or inside other organisms. Plants also have their own viruses. Some plant viruses can spread through insects and damage crops like tomatoes, potatoes, and wheat. This ability to infect so many different life forms shows how adaptable viruses are. Each type of virus is usually designed to target very specific hosts, almost like a key that only fits certain locks. This specialization allows viruses to exist in nearly every ecosystem on the planet. Some viruses don't infect humans, animals, or plants at all. Instead, they infect bacteria. These special viruses are called bacteriophages, often shortened to simply phages. While that sounds dramatic, it actually describes what they do quite well. A bacteriophage attaches to a bacterium, injects its genetic material, and then forces the bacterial cell to produce new viruses. Eventually, the bacterium bursts open, releasing many new phages. Bacteriophages have a very unusual shape. Many look like tiny lunar landers, with a geometric head, a tail, and thin leg-like fibers used to grab onto bacteria. They are incredibly common and play an important role in nature by controlling bacterial populations. Without bacteriophages constantly infecting bacteria, some bacterial populations could grow out of control in environments like oceans and soil. Bacteriophages are not just one type of virus. They are actually the most common viruses on Earth. Scientists believe they make up a huge portion of the trillions upon trillions of viruses that exist on the planet. Their success comes from one simple reason: bacteria are everywhere. Bacteria live in oceans, soil, inside animals, and even deep underground. Wherever bacteria exist, bacteriophages are usually there too, constantly infecting and controlling bacterial populations. In the oceans alone, phages destroy billions of bacteria every second. This may sound destructive, but it actually helps keep ecosystems balanced by preventing certain bacteria from taking over. A helpful way to picture this is to imagine a natural population control system. Just as predators keep animal populations in check, bacteriophages help regulate bacterial numbers, playing a quiet but vital role in maintaining the balance of life on Earth. Viruses are found in almost every environment on Earth, from the deepest oceans to the highest parts of the atmosphere. They exist in places where humans live, but also in environments where life seems nearly impossible. Scientists have discovered viruses in deserts, polar ice, deep underground rock layers, and even inside volcanic hot springs. Some have also been detected high in the atmosphere, where winds can carry them thousands of kilometers across the planet. Many people think viruses mainly exist around sick people or hospitals, but that's a misunderstanding. In reality, viruses are a natural and constant part of Earth's ecosystems. They live in soil, water, plants, animals, and microbes. Every ecosystem, from forests to coral reefs, contains its own viral communities. Because viruses infect many different types of organisms, they play an important role in shaping populations and maintaining balance in nature. Viruses exist in nearly every place you can imagine. Scientists have found them in oceans, soil, the air, and inside almost all living organisms. In the oceans, viruses infect tiny microbes that form the base of the marine food chain. In soil, they interact with bacteria and fungi that help break down organic material. Even the air around us can contain virus particles drifting with dust and microscopic droplets. Your own body also contains viruses. In fact, the human body hosts trillions of viruses, most of which infect bacteria living in your gut, rather than your own cells. These viruses are part of a complex ecosystem called the human microbiome. Many people think viruses only appear when someone gets sick, but in reality, viruses are constantly present in the environment, quietly interacting with living organisms and ecosystems all around us. A single teaspoon of seawater may contain millions of viruses. That tiny amount of water, about the size of a spoon you'd use for sugar in coffee, holds an invisible world filled with microscopic virus particles. Most of these viruses infect marine microbes, especially bacteria and tiny plankton. These microorganisms are extremely important because they form the foundation of ocean ecosystems, supporting fish, whales, and many other marine animals. When viruses infect these microbes, they often destroy them. While that might sound harmful, it actually helps keep the ocean balanced. By controlling microbial populations, viruses prevent certain species from growing too quickly. Scientists estimate that viruses in the oceans destroy billions of microbes every day. This process releases nutrients back into the water, helping feed other organisms. So in one small spoon of seawater, there may be more viruses than people on Earth. Viruses are incredibly tiny, far smaller than most bacteria. In fact, many viruses are about 10 to 100 times smaller than a typical bacterial cell. To picture this difference, imagine a basketball compared to a grain of sand. That's roughly the scale difference between many bacteria and viruses. Because of their tiny size, viruses cannot be seen with regular microscopes that can easily show bacteria. Early scientists were confused by this. In the late 1800s, researchers noticed that certain infectious agents could pass through filters that normally trapped bacteria. This mysterious substance turned out to be viruses. Their small size is also part of what makes viruses so effective. Being so tiny allows them to enter cells easily and spread quickly through microscopic spaces in tissues and environments. Even though they are extremely small, their impact on living organisms can be enormous. Most viruses are unbelievably small, typically measuring between 20 and 300 nanometers in width. A nanometer is 1/1000th of a meter, which is far beyond what the human eye can see. To help visualize this, consider a human hair, which is about 80,000 to 100,000 nanometers wide. Even a large virus at 300 nanometers would still be hundreds of times thinner than a single strand of hair. Because of their tiny size, viruses cannot be seen with ordinary microscopes. Scientists need powerful electron microscopes, which use beams of electrons instead of light, to observe them clearly. Many people imagine viruses as large germs floating around like bacteria, but that's not accurate. Viruses are more like ultra-microscopic particles, almost at the boundary between chemistry and biology. Despite their minuscule size, these tiny structures can still infect cells and multiply rapidly. Viruses are so incredibly small that thousands of them could fit across the width of a single human hair. A typical human hair is about 80,000 to 100,000 nanometers wide, while many viruses are only 50 to 100 nanometers across. If you lined viruses side by side across one strand of hair, you could fit hundreds to thousands of them in a single row. Imagine placing tiny grains of dust across a thick rope. That's roughly the scale difference. This extreme smallness is one reason viruses remained unknown for so long. Early microscopes simply could not see them, which is why scientists originally suspected that mysterious infections were caused by toxins rather than actual particles. Today, powerful electron microscopes reveal that viruses have intricate shapes and structures, despite being incredibly tiny. It's amazing that something so small can still invade cells and replicate in huge numbers. The word virus comes from an old Latin word meaning poison or toxic fluid. When early scientists first studied infectious diseases, they didn't know that viruses were tiny particles. They simply noticed that certain diseases seemed to spread through something invisible, because these illnesses behaved like a mysterious poison that could move from one organism to another. Scientists used the word virus to describe the unknown cause. At the time, people imagined viruses as some kind of toxic substance, not as microscopic structures with genetic material. It wasn't until much later, with the development of advanced microscopes and laboratory techniques, that researchers realized viruses were actually tiny biological particles. Interestingly, the original name still makes sense in a way. Even though viruses are not literally poison, many of them can disrupt cells and cause disease, which likely reminded early scientists of toxic effects. Viruses were first discovered in the late 1800s, long before scientists could actually see them. At the time, researchers were trying to understand diseases that affected plants and animals, but something strange kept happening. Certain infectious agents could pass through special filters designed to trap bacteria. Normally, bacteria are large enough to be caught by these filters, so scientists expected the infection to stop there. But in some cases, the filtered liquid could still infect healthy plants. This meant the disease-causing agent had to be much smaller than bacteria, so small that it slipped through the filter. One of the earliest examples involved a plant disease affecting tobacco leaves. Scientists realized that something invisible was spreading the illness, even though no bacteria could be found. This mysterious infectious particle later became known as a virus, opening the door to an entirely new field of science. When viruses were first discovered in the late 1800s, many scientists believed they were toxic chemicals rather than living particles. This misunderstanding happened because viruses were too small to be seen with the microscopes available at the time. Researchers knew that something was causing infections, but they couldn't see any bacteria or organisms responsible for the disease. The infectious substance could also pass through filters that normally trapped bacteria. Because of this, scientists assumed the illness must be caused by a poisonous fluid or toxin produced by microbes. It wasn't until decades later, with the development of more advanced laboratory techniques and eventually electron microscopes, that scientists realized viruses were actually tiny particles containing genetic material. This discovery completely changed our understanding of infectious disease and biology, revealing that viruses are unique biological entities, not just invisible poisons. The first virus ever discovered was the tobacco mosaic virus, a virus that infects tobacco plants. Scientists identified it in 1892 while studying a strange disease that caused tobacco leaves to develop green and yellow mosaic-like patterns. At first, researchers thought bacteria were responsible. But when infected plant sap was passed through filters designed to trap bacteria, the liquid could still infect healthy plants. This meant the disease was caused by something much smaller than bacteria. This mysterious agent became known as the tobacco mosaic virus, making it the first virus ever recognized by science. Later, scientists used powerful electron microscopes to finally see the virus. It turned out to have a long rod-like shape, almost like a microscopic tube. The discovery of this plant virus opened the door to the entire field of virology, the scientific study of viruses. Viruses can mutate extremely quickly, meaning their genetic instructions can change in a short amount of time. These mutations happen when viruses copy their genetic material while making new virus particles. Unlike many living organisms that have complex systems to correct mistakes in their DNA, many viruses lack strong error-checking mechanisms. As a result, small copying errors occur frequently during replication. To visualize this, imagine someone repeatedly copying a long sentence by hand thousands of times. Eventually, small spelling mistakes start to appear. The same thing happens when viruses replicate. Most mutations don't make much difference, but occasionally a change can help a virus spread more easily, infect new hosts, or avoid the immune system. This rapid mutation is one reason viruses can adapt so quickly. It also explains why certain viral diseases may change from year to year, making them harder to control. Because viruses mutate so quickly, they can adapt to new hosts and environments surprisingly fast. Each time a virus replicates, small genetic changes may appear. Most of these changes do nothing useful, but occasionally one gives the virus an advantage. For example, a mutation might help a virus attach more easily to a cell, reproduce faster, or survive better inside a new species. If that change helps the virus spread, it can quickly become common in the viral population. You can think of this like trying thousands of keys in a lock. Eventually, one key might fit. In the same way, viral mutations sometimes produce a version that can infect a new host. This ability to adapt explains why viruses can sometimes jump from animals to humans. It also helps explain why new viral strains can appear over time, especially during large outbreaks. Not all viruses mutate at the same speed. RNA viruses usually mutate much faster than DNA viruses. The reason has to do with how their genetic material is copied. RNA viruses use special enzymes to copy their RNA, but these enzymes often lack strong proofreading abilities. That means mistakes happen frequently when the virus replicates. Each copying error becomes a mutation. DNA viruses, on the other hand, often have better error-correcting systems, similar to those used by many living cells. Because of this, their genetic material tends to remain more stable over time. To imagine the difference, think of two people copying a long paragraph. One carefully checks every sentence, while the other copies quickly without proofreading. The fast writer will naturally make more mistakes. This is why many well-known viruses with RNA genomes can change rapidly, sometimes creating new strains over time. Some viruses have the ability to jump from one species to another. This means a virus that normally infects animals can sometimes adapt and begin infecting humans. At first, the virus may not spread easily in the new species, but because viruses mutate quickly, a small genetic change can sometimes allow the virus to attach to cells in a different host. You can think of it like a key that almost fits a lock. With a few small changes, it may suddenly work perfectly. This kind of cross-species jump has happened many times throughout history. Many well-known human diseases originally came from animals before adapting to humans. People sometimes assume viruses are designed specifically for humans, but that's rarely true. In most cases, humans are actually new hosts, and the virus evolved somewhere else before eventually making the jump. When a virus moves from animals into humans, the process is called zoonotic transmission. The word zoonotic comes from Greek roots meaning animal disease. This happens when a virus that normally lives in animals gains the ability to infect people. Often the virus has been circulating in animals for a long time before the jump occurs. Through mutations or close contact between species, the virus eventually finds a way to enter human cells. A good way to imagine this is like a password system. Animal viruses usually have the correct password to enter animal cells. Occasionally, a mutation changes that password just enough to also unlock human cells. Zoonotic transmission is actually quite common in nature. Many viruses circulate quietly in wildlife populations and only rarely spill over into humans. When they do, scientists study them closely to understand how they adapted to a new host. Many diseases that affect humans today actually began in animals before eventually adapting to people. This happens because viruses often circulate naturally in wildlife or livestock long before humans become infected. When humans come into close contact with animals through farming, hunting, or shared environments, there is sometimes an opportunity for a virus to cross the species barrier. If the virus can successfully infect human cells and spread between people, it may eventually become a human disease. You can imagine this like a guest entering a new house for the first time. At first, the guest may struggle to find their way around, but with small adjustments, they can learn how everything works. Scientists estimate that more than half of emerging infectious diseases in humans originated in animals. This is why monitoring viruses in wildlife populations is an important part of preventing future outbreaks. For a virus to infect a cell, it first has to attach to special molecules on the cell's surface called receptors. These receptors act like tiny docking points. A useful way to picture this is with a lock and key system. The virus carries structures on its surface that function like keys, while the receptors on the cell act like locks. If the key fits the lock, the virus can attach and begin the infection process. If the key doesn't fit, the virus simply cannot enter the cell. This is one reason viruses often infect only certain types of cells. For example, some viruses target cells in the lungs, while others prefer nerve cells or cells in the liver. It also explains why certain species are resistant to particular viruses. If the right receptor isn't present, the virus has no doorway into the cell. Most viruses are very picky about the cells they infect. Each type of virus usually targets specific cells in the body, depending on which receptors it can recognize and attach to. You can think of this like a delivery service that only goes to certain addresses. Even though the virus may travel through the body, it can only enter cells that have the correct surface receptors. For example, some viruses mainly infect cells in the respiratory system, which is why they cause symptoms like coughing or breathing problems. Others target nerve cells, skin cells, or cells in the liver. This is also why different viruses cause very different diseases. The symptoms often depend on which cells or organs are being infected. If a virus cannot find the right type of cell with the correct receptor, it simply cannot continue its life cycle. Many viruses are extremely specialized and can infect only a single species. This happens because viruses rely on very specific receptors and cellular conditions to enter and reproduce inside cells. You can imagine this like a key that fits only one exact lock. Even small differences between species can prevent a virus from attaching to cells. If the key doesn't match the lock, the virus simply cannot get inside. For example, some viruses infect only certain animals or plants, and they cannot infect humans at all. Even closely related species can be protected if their cell receptors are slightly different. This specialization is one reason many viruses remain confined to particular hosts for long periods of time. However, occasional mutations may change the key, allowing a virus to infect a new species, which is how cross-species infections sometimes begin. While many viruses infect only one species, some viruses are much less selective and can infect a wide range of hosts. These viruses are able to recognize receptors that exist in multiple species, allowing them to cross between animals more easily. You can think of this like a master key that opens several different locks instead of just one. Because these viruses can interact with similar cell receptors in different species, they have more opportunities to spread. Viruses with a broad host range may infect several types of animals, and sometimes humans as well. This ability can make them more likely to jump between species, especially when animals and humans live in close contact. However, infecting multiple hosts is still difficult. Even small differences in cell biology can stop a virus from spreading effectively. When a virus does succeed in multiple species, scientists pay close attention because it can increase the chances of new outbreaks. Viruses cannot build new virus particles by themselves. Instead, they take control of a host cell's machinery and force it to do the work for them. Inside every cell are tiny biological machines that normally produce proteins and other materials needed for the cell to survive. When a virus enters the cell, it releases its genetic instructions and reprograms these machines. It's similar to a factory takeover. Imagine a factory that normally produces cars. Suddenly, someone replaces the blueprints, and the factory begins producing an entirely different product. In the same way, the infected cell stops making its normal components and starts assembling virus parts instead. These parts are then combined to create complete virus particles. Eventually, the cell may release hundreds or even thousands of new viruses, each capable of infecting another cell. Once a virus successfully enters a cell, one of the first things it does is copy its genetic material. This process is called replication, and it allows the virus to produce many new copies of itself. The virus releases its DNA or RNA inside the host cell. Then it uses the cell's own molecular machinery to duplicate those genetic instructions again and again. Each copy will later become the blueprint for a new virus particle. You can imagine this like photocopying a document thousands of times. The original instructions enter the cell, and soon the cell is filled with copies. Without this step, viruses would not be able to spread. Replicating the genetic material is essential because every new virus needs its own set of instructions. After replication finishes, the cell begins assembling the copied genetic material into new virus particles, preparing them to infect other cells. Once a virus takes over a cell, the cell can start producing huge numbers of new virus particles. In many cases, a single infected cell can generate thousands of viruses before the infection cycle ends. After the virus replicates its genetic material and builds new protein shells, the cell begins assembling complete virus particles. These new viruses fill the cell like products, stacking up in a factory warehouse. Imagine a tiny factory that suddenly switches production and starts rapidly producing thousands of identical items. That's essentially what happens inside an infected cell. Eventually, the cell becomes so packed with virus particles that it can no longer function properly. At that point, the viruses are ready to leave the cell and move on to infect other nearby cells. This rapid multiplication is one reason viral infections can spread quickly inside the body once they begin. In many viral infections, the virus eventually leaves the host cell by bursting it open. This process is called cell lysis. After the infected cell produces hundreds or thousands of new virus particles, the inside of the cell becomes extremely crowded. Eventually, the cell membrane can no longer hold everything inside. The cell breaks apart, releasing all the newly made viruses at once. You can imagine it like a balloon filled with too much air suddenly popping. When it bursts, everything inside is released instantly. This sudden release allows the viruses to quickly spread and infect nearby cells. However, it also means the original cell is completely destroyed. Many bacteriophages, viruses that infect bacteria, use this method. By bursting their host cells, they release large numbers of viruses that can immediately begin infecting new bacterial cells nearby. Not all viruses destroy the cells they infect. Some viruses can exit a cell without immediately killing it. Instead of bursting the cell open, they slowly leave by passing through the cell's outer membrane. This process is often called budding. As new virus particles form inside the cell, they push outward and wrap themselves in a small piece of the cell's membrane before leaving. You can picture this like bubbles gently forming and floating away from the surface of water. The cell remains intact while the viruses gradually escape. Because the cell is not destroyed right away, it may continue producing viruses for some time. This allows the infection to spread more slowly but continuously. Some viruses that infect animals use this method. It helps them spread while keeping the host cell alive long enough to keep producing more virus particles. Not all viruses begin multiplying right away after infecting a host. Some viruses can enter a dormant state, meaning they stay inside the body but remain inactive for long periods of time. During dormancy, the virus is essentially sleeping. It is present in certain cells, but it is not actively producing new virus particles or causing symptoms. This state can sometimes last months, years, or even decades. A helpful way to picture this is like a computer program running quietly in the background without doing anything noticeable. The virus is still there, but it isn't actively causing trouble. Later, certain triggers, such as stress, illness, or changes in the immune system, can cause the virus to reactivate. When that happens, the virus begins replicating again, and symptoms may appear. This ability to hide inside the body makes some viral infections very difficult to completely eliminate. Some viruses that enter a dormant state don't stay inactive forever. Under certain conditions, they can reactivate and begin reproducing again, sometimes long after the original infection happened. This means a person may carry a virus quietly in their body for years without noticing it. Then something changes, such as stress, illness, fatigue, or a weakened immune system, and the virus becomes active again. You can think of this like a campfire that appears to be out, but still has hot embers underneath. When fresh oxygen or fuel is added, the fire can suddenly flare up again. When a dormant virus reactivates, it may begin producing new virus particles and infecting nearby cells. This can lead to symptoms returning, even though the original infection may have happened a long time ago. This reactivation ability is one reason certain viral infections can reappear throughout a person's life. Some viruses have an unusual hiding place inside the body. Herpes viruses, for example, can hide inside nerve cells, where they may remain dormant for long periods of time. After the initial infection, the virus travels along nerve pathways and settles inside nerve cells near the spine or face. There, it can stay inactive for months or even years without causing symptoms. You can imagine this like a traveler quietly staying in a hidden cabin deep in the forest. The virus remains there, unnoticed, waiting for the right conditions to become active again. Later, certain triggers, such as stress, illness, sunlight, or fatigue, can wake the virus up. When this happens, it travels back along the nerve to the skin, where it can cause symptoms again. This is why infections caused by herpes viruses can come and go repeatedly throughout a person's life. Some viruses go even further than simply hiding inside a cell. They can actually insert their genetic material directly into the host's DNA. When this happens, the virus becomes part of the cell's genetic instructions. The cell may continue living and dividing normally, but the viral DNA remains quietly embedded within its genome. You can think of this like adding an extra page into a large instruction manual. The manual still works, but now it contains new instructions that were not originally there. Because the viral DNA is now part of the cell's genome, it can sometimes be copied every time the cell divides. This allows the viral genetic material to persist for a long time, even if the virus is not actively producing new particles. This strategy helps certain viruses stay hidden in the body and avoid immediate detection by the immune system. When a virus inserts its genetic material into the DNA of a host cell, the viral DNA becomes what scientists call a provirus. In this state, the virus is essentially hidden inside the cell's genetic instructions. The host cell may continue functioning normally, but the viral DNA is quietly sitting there as part of the genome. A good way to picture this is like a hidden paragraph inserted into a long instruction manual. Most of the time, the manual runs as usual, but that extra paragraph can be activated later. When the provirus becomes active, it can begin producing new virus particles using the cell's machinery. In some cases, the provirus can remain inactive for a very long time before anything happens. Because the viral DNA is copied whenever the cell divides, this strategy allows the virus to persist inside the body for years or even a lifetime. It may sound surprising, but a portion of the human genome actually comes from ancient viruses. Over millions of years, certain viruses inserted their genetic material into the DNA of our ancestors' cells. Normally, this viral DNA would disappear when infected cells die. But in rare cases, the virus inserted its genes into reproductive cells, such as sperm or egg cells. When that happened, the viral DNA could be passed down to future generations. You can think of it like an old message accidentally written into a family recipe book and copied every time the book is reprinted. Over many generations, that message becomes part of the book itself. Because of this process, fragments of viral DNA have been inherited for millions of years. Today, scientists estimate that a noticeable portion of human genetic material contains remnants of these ancient viral infections. About 8% of the human genome is made of genetic material that originally came from viruses. That may sound shocking, but it means that nearly 1 out of every 12 pieces of human DNA has viral origins. To visualize this, imagine a large instruction book containing 100 pages. Around 8 of those pages would contain leftover instructions from ancient viruses that infected our distant ancestors millions of years ago. These viral sequences entered the human genome when certain viruses inserted their DNA into reproductive cells, allowing the genetic material to be passed down through generations. Most of these viral fragments are no longer active. They are essentially genetic fossils, pieces of ancient viruses that no longer function as viruses. Even though they are inactive, their presence shows that viruses have been interacting with life on Earth for millions of years, leaving permanent marks in our genetic history. The viral DNA that became part of the human genome is known as endogenous retroviruses, often shortened to ERVs. The word endogenous means something that originates from within the body. These sequences are the remains of ancient retroviruses that infected our distant ancestors millions of years ago. Retroviruses are a special type of virus that can insert their genetic material directly into the DNA of a host cell. When this happened in reproductive cells, the viral DNA became permanently inherited, passing from parent to child across generations. You can think of endogenous retroviruses as genetic fossils hidden inside our DNA. Just like fossils in rock reveal ancient life forms, these viral sequences reveal infections that occurred long before humans even existed. Today, scientists can identify thousands of these viral fragments scattered throughout the human genome, providing clues about ancient viral encounters in evolutionary history. Although many viral fragments in human DNA are inactive, some have actually been repurposed by our bodies for useful functions. In other words, genes that originally came from viruses have been adapted to help human biology. This might sound strange, but evolution sometimes reuses useful genetic tools, even if they originally came from viruses. You can imagine this like finding an old machine part and discovering it works perfectly in a completely different device. Instead of throwing it away, the system keeps using it. Over millions of years, some viral genes were modified and integrated into normal biological processes. Instead of producing viruses, they now help cells perform specific tasks. Scientists are still discovering how many of these viral genes have been reused by our bodies. This surprising relationship shows that viruses have not only caused disease throughout history, they have also influenced how complex life evolved. One of the most surprising examples of viral DNA helping humans involves the formation of the placenta during pregnancy. Scientists discovered that a key gene involved in building the placenta originally came from an ancient virus. This gene helps certain cells in the developing placenta fuse together, creating a special layer that allows nutrients and oxygen to pass from the mother to the baby. To imagine its importance, think of the placenta as a life support bridge between mother and child. Without it, the growing baby would not receive the oxygen and nutrients needed to develop. The gene responsible for this process is believed to have originated from an ancient viral infection that inserted its DNA into the genome of our distant ancestors millions of years ago. Over time, evolution kept the gene because it turned out to be extremely useful for reproduction. Viruses don't just infect organisms. They can also move genes from one species to another, helping drive evolution over time. When a virus infects a cell, it sometimes accidentally picks up pieces of the host's genetic material. Later, if that virus infects a different organism, it may deliver that genetic fragment into the new host's DNA. You can imagine this process like a courier accidentally carrying pages from one book into another book. Over time, those extra pages can introduce new information. Although this doesn't happen all the time, it has occurred often enough throughout history to influence how life evolves. Certain genes found in different species may have spread, partly because viruses carried them. This means viruses have acted like tiny genetic messengers, occasionally transferring useful traits and helping shape the diversity of life on Earth. When viruses move genetic material between different organisms, the process is called horizontal gene transfer. The word horizontal means the genes move across different organisms rather than passing normally from parent to child. Under normal circumstances, genetic information flows vertically, meaning parents pass genes to their offspring. But horizontal gene transfer works differently. It allows genes to move between unrelated organisms, sometimes even between completely different species. You can imagine this like borrowing pages from someone else's instruction manual, instead of inheriting the manual from your parents. Viruses can make this possible because they enter cells and interact directly with genetic material. Occasionally, they carry pieces of DNA from one organism into another. This process has played a role in the evolution of many microbes and may even have influenced some larger life forms over long periods of time. Viruses don't just affect individual organisms. They can influence entire ecosystems. Because viruses infect many different forms of life, they play an important role in controlling populations of microbes, plants, and animals. In many environments, especially the oceans, viruses constantly infect microorganisms, such as bacteria and plankton. These microbes are the foundation of many food chains, supporting larger organisms like fish and marine mammals. You can imagine viruses acting like natural regulators in an ecosystem, similar to how predators keep animal populations balanced. When viruses infect and destroy certain microbes, they prevent those populations from growing too large. This helps maintain diversity among different species. Without viruses performing this role, some microbial populations could dominate entire environments, which might disrupt ecosystems. In this way, viruses quietly help maintain balance in nature, even though they are invisible to us. The oceans are filled with viruses, and many of them infect microscopic organisms that live in seawater. These marine viruses help regulate the populations of ocean microbes, especially bacteria and tiny plankton. Microbes in the ocean multiply very quickly, without something limiting their numbers. Some species could grow so fast that they overwhelm others. Viruses help prevent this by infecting and destroying certain microbes, keeping populations balanced. You can think of this like natural population control in a crowded city. If one group grows too quickly, something steps in to keep things balanced. This process is extremely important because ocean microbes are responsible for producing a large portion of Earth's oxygen and supporting marine food webs. Fish, whales, and many other animals ultimately depend on them. By controlling microbial populations, marine viruses quietly help maintain the health and stability of ocean ecosystems. Viruses destroy billions of microbes every single day, especially in the world's oceans. In fact, scientists estimate that viruses kill an enormous number of bacteria and microscopic plankton every second. At first, this may sound harmful, but it actually plays an important role in keeping ecosystems balanced. Without viruses limiting their numbers, many microbes could multiply out of control, crowding out other species. You can picture this like pruning plants in a garden. Removing some plants helps the rest of the garden stay healthy and balanced. When viruses destroy these microbes, the contents of the cells, such as nutrients and organic molecules, are released back into the surrounding water. These nutrients then become available to other microorganisms. This process helps recycle important materials in the ocean, supporting the marine food web that eventually sustains fish, whales, and many other sea creatures. When viruses infect and destroy microbes in the ocean, they trigger an important process that releases nutrients back into the water. When a microbial cell bursts open, everything inside it, carbon, nitrogen, and other molecules, spills into the surrounding seawater. You can think of this like breaking open a tiny nutrient packet. The materials that were locked inside the cell suddenly become available to other organisms. This process is often called the viral shunt, and it plays a huge role in ocean ecosystems. Instead of nutrients being locked inside living microbes, they get recycled quickly and reused by other microscopic life. Since the ocean contains trillions of microbes, this constant recycling happens on an enormous scale every day. Without viral infections releasing these nutrients, many essential elements would stay trapped inside microbial cells, and the flow of nutrients through the ocean's food web would be much slower. The nutrients released when viruses destroy ocean microbes don't just disappear, they become fuel for the entire marine food web. Tiny organisms quickly absorb these recycled nutrients and use them to grow. These microscopic organisms, such as bacteria and plankton, form the foundation of ocean life. Small marine animals feed on them. Larger fish feed on those animals, and eventually predators like sharks, seals, and whales depend on the same chain. You can imagine this system like recycling in a city. Waste materials are collected, processed, and reused to create new resources. In the ocean, viruses help break down microbes so their nutrients can be reused again and again. Because oceans cover about 70% of Earth's surface, this recycling process happens on a massive scale. Viral activity helps keep nutrients moving through the ecosystem, supporting the life of countless marine species. In certain situations, viruses can actually help prevent environmental problems by controlling harmful bacterial blooms. A bacterial bloom happens when bacteria multiply very quickly and form dense populations in water. Some blooms can become dangerous because they produce toxins or consume large amounts of oxygen, which can harm fish and other aquatic life. This is especially common in lakes, rivers, and coastal waters. Viruses that infect bacteria can act like natural regulators in these situations. When bacterial populations grow too large, viruses begin infecting those bacteria and destroying many of them, slowing the bloom. You can imagine this like a natural reset button. that prevents one group of organisms from taking over the entire ecosystem. While viruses are often seen as harmful, in cases like this they can actually help stabilize ecosystems by preventing certain bacterial populations from growing out of control. Viruses don't just affect microbes in the short term, they can also shape how microbes evolve over time. Because viruses constantly infect bacteria and other microorganisms, they create strong pressure for those microbes to adapt and develop defenses. When a virus infects a microbial population, many cells may be destroyed. However, a few microbes might have small genetic differences that help them resist the infection. These survivors continue reproducing, passing their protective traits to future generations. You can imagine this like a survival challenge, where only the organisms with the right defenses make it through. Over many generations, this process can lead to new microbial traits and adaptations. In response, viruses may also evolve new ways to infect their hosts. This ongoing back and forth creates what scientists call an evolutionary arms race, constantly shaping both viruses and the microbes they infected. Scientists estimate that viruses destroy about 20% of all microbes in the ocean every single day. That means roughly 1 out of every 5 microbial cells in seawater is infected and killed by viruses daily. At first, this might sound like a disaster for ocean life, but surprisingly, this constant viral activity is actually essential for maintaining balance in marine ecosystems. Imagine a huge city where the population grows extremely fast. Without some form of control, resources could quickly run out. In the ocean, viruses act like natural population regulators, preventing certain microbial species from becoming too dominant. By constantly infecting and destroying microbes, viruses keep microbial populations diverse and balanced. At the same time, the destroyed cells release nutrients back into the water. This daily cycle happens across the entire ocean, quietly influencing the health of Earth's largest ecosystem. Although viruses are often associated with disease, some viruses can actually be helpful to humans. Not every virus causes harm. In fact, scientists are discovering that certain viruses can be used in medicine and biotechnology. For example, some viruses infect bacteria rather than human cells. These viruses can help control harmful bacteria, which may reduce infections or help balance microbial communities. Viruses are also used by researchers as biological tools. Because they are naturally good at entering cells and delivering genetic material, scientists can modify viruses to carry helpful genes into cells for medical treatments. You can think of these viruses like tiny delivery vehicles, designed to transport genetic instructions exactly where they are needed. This challenges the common belief that all viruses are dangerous. While many viruses cause disease, others are now being studied as valuable tools for science and medicine. Surprisingly, certain viruses are being used to treat bacterial infections. These viruses are called bacteriophages, and they specifically infect and destroy bacteria. Since bacteriophages only target bacteria, they leave human cells completely unharmed. This makes them potentially useful for fighting harmful bacteria that cause infections. You can imagine them like precision-guided hunters that search for a specific type of bacterium, attach to it, and destroy it. Once the bacteria burst, the newly released phages can move on to attack other bacterial cells. This approach is especially interesting because some bacteria have become resistant to antibiotics. In those cases, bacteriophages may offer another way to fight infections. Scientists are currently researching how phages can be used safely and effectively in medicine. Although the idea sounds futuristic, these viruses are actually natural bacteria-killing machines that already exist in the environment. Using bacteriophages to treat bacterial infections is known as phage therapy. In this approach, doctors use specific viruses that infect and destroy harmful bacteria inside the body. The idea is simple. Instead of using chemical antibiotics, Phage therapy uses naturally occurring viruses as targeted bacterial killers. Each type of bacteriophage usually attacks only one or a few specific bacteria, making the treatment very precise. You can think of it like sending trained search and destroy agents after a particular bacterial target. Once the phage finds the right bacterium, it infects the cell, multiplies inside it, and eventually destroys it. One advantage of phage therapy is that the viruses can multiply at the infection site. As long as the harmful bacteria are present, the phages continue reproducing and attacking them. Because antibiotic resistance is becoming a major problem worldwide, scientists are increasingly studying phage therapy as an alternative medical treatment. Phage therapy is not a new idea. In fact, it was used before antibiotics were discovered. Scientists began studying bacteriophages in the early 1900s, and doctors soon realized these viruses might be useful for treating bacterial infections. At the time, researchers experimented with using phages to treat illnesses such as dysentery and other bacterial diseases. In some places, doctors successfully used these virus-based treatments to help patients recover. However, when antibiotics like penicillin were discovered in the 1940s, they quickly became the dominant treatment for bacterial infections. Antibiotics were easier to produce and use, so phage therapy gradually faded from mainstream medicine in many countries. But the idea never completely disappeared. In some parts of the world, phage therapy continued to be researched and used for decades. Today, scientists are revisiting this older approach as a possible solution to antibiotic-resistant bacteria. In recent years, phage therapy has been gaining renewed attention from scientists and doctors. One major reason is the growing problem of antibiotic resistance. Some bacteria have evolved defenses that make certain antibiotics less effective or even useless. Because bacteriophages naturally attack bacteria, researchers are exploring whether these viruses could become alternative treatments for difficult infections. Unlike antibiotics, which often kill many types of bacteria at once, phages usually target very specific bacterial species. You can imagine this like using a precision tool instead of a wide spray. The phage goes directly after the harmful bacteria while leaving many helpful microbes untouched. Modern technology is also helping scientists study and customize bacteriophages more carefully than before. As research continues, phage therapy may become an important tool in the future fight against drug-resistant bacterial infections. Viruses are not all shaped the same. In fact, they come in many different shapes and structures, which can look surprisingly complex when viewed under powerful electron microscopes. Some viruses appear round or spherical, others look like long rods or thin filaments, and some have shapes that resemble tiny machines. Their structure depends mainly on how their protein shell, called the capsid, is built. You can imagine viruses like different styles of containers, each designed to carry and protect genetic material in its own way. These shapes are not random. The structure helps the virus attach to cells, protect its genetic instructions, and sometimes deliver them into the host cell. Certain shapes also make the virus more stable in different environments. Even though viruses are incredibly small, thousands could fit across a human hair, they can have remarkably precise and organized structures. Many viruses have a spherical or nearly round shape when viewed under powerful electron microscopes. These viruses appear like tiny microscopic balls, even though they are thousands of times smaller than anything the human eye can see. The spherical appearance comes from the way the virus's protein shell, called the capsid, is arranged. The proteins form a structured outer layer that surrounds and protects the virus's genetic material. To visualize the scale, imagine a perfectly round soccer ball, but shrunk down so small that thousands of them could fit across the width of a single human hair. This spherical design is not just for appearance. The structure helps the virus protect its genetic instructions and maintain stability while traveling between host cells. Many well-known viruses that infect animals and humans have this general round shape, making it one of the most common virus structures in nature. Not all viruses are round. Some viruses have long, rod-like or filament shapes, making them look more like tiny threads or tubes when viewed under an electron microscope. Instead of forming a spherical shell, the virus's proteins arrange themselves into a long spiral structure that wraps around the genetic material. This creates a shape similar to a microscopic straw or noodle. To visualize the difference, imagine comparing a soccer ball to a piece of spaghetti. Both can hold material inside, but their shapes are completely different. Certain plant viruses are well known for this rod-like structure. Their long, narrow form can help them move through the tiny channels inside plant cells. These filament shapes also show how flexible viral design can be. Even though viruses are extremely simple compared to cells, their structures can vary greatly, depending on how their protective protein shells are assembled. Some bacteriophages have one of the most unusual shapes in the microscopic world. When seen under an electron microscope, they can look like tiny robotic machines or miniature lunar landers. These viruses often have a geometric head, which holds their genetic material, connected to a long tail structure. At the end of the tail are thin fibers that act like legs, allowing the virus to attach firmly to a bacterium. You can imagine the process like a spacecraft landing on a planet. The virus lands on the surface of a bacterium, anchors itself with the tail fibers, and then injects its genetic material into the cell. This injection works somewhat like a microscopic syringe, delivering viral instructions directly into the bacterium. Despite their mechanical appearance, these structures form naturally through simple protein assembly, creating one of the most recognizable and fascinating virus shapes scientists have discovered. The shape of a virus is mainly determined by its protein shell, which surrounds and protects the virus's genetic material. This shell is built from many small protein pieces that naturally fit together in specific patterns. When these proteins assemble, they create the virus's overall structure, whether it ends up looking spherical, rod-shaped, or like a tiny machine. You can imagine this process like building something with identical Lego blocks. Depending on how the blocks are arranged, you can create very different shapes, even though the pieces are the same. This protein shell does more than just determine the virus's appearance. It also helps protect the virus's genetic instructions, survive in different environments, and sometimes assist in attaching to host cells. Because the shell must be strong yet efficient, viral proteins often assemble into highly organized geometric patterns that maximize stability while using very little material. The protective outer shell of a virus is called a capsid. This structure surrounds the virus's genetic material and helps keep it safe while the virus travels between host cells. A capsid is built from many repeating protein units that fit together in very precise ways. These proteins automatically assemble into a strong outer shell without needing complex machinery. You can think of the capsid like a protective container or armor that carries the virus's genetic instructions. Just as a helmet protects a person's head, the capsid protects the virus's DNA or RNA from damage. The capsid also plays another important role. In many viruses, parts of the capsid help the virus recognize and attach to specific cells before infection begins. Despite being incredibly tiny, often only a few hundred nanometers wide, the capsid is a highly organized structure built with remarkable efficiency. The capsid's most important job is to protect the virus's genetic material. Inside every virus is DNA or RNA containing the instructions needed to produce new virus particles. Without protection, this delicate genetic material could easily be damaged by heat, chemicals, or enzymes in the environment. The capsid acts like a strong protective case, similar to how a phone case protects a smartphone from scratches or impacts. Even though the virus itself is extremely tiny, the capsid forms a tough outer layer that keeps the genetic instructions intact. This protection is especially important when viruses travel outside a host cell, where conditions can be harsh. Some viruses may survive on surfaces, in water, or in the air before finding a new host by shielding the genetic material until infection occurs. The capsid ensures the virus's instructions remain ready to activate once it enters a new cell. Many viral capsids are built with remarkably precise geometric symmetry. Instead of random shapes, the proteins that form the capsid arrange themselves into organized patterns that repeat over and over. You can imagine this like a soccer ball, where identical panels fit together to create a strong, balanced structure. In viruses, the repeating protein units assemble in similar geometric arrangements, forming highly stable shells. This symmetry is very efficient. By repeating the same small protein many times, the virus can build a large protective shell using minimal genetic instructions. Since viruses have limited genetic material, this strategy allows them to create strong structures without needing many different proteins. Scientists often admire viral capsids because of their mathematical elegance. Even though viruses are incredibly tiny, far smaller than bacteria, their protein shells form some of the most beautifully organized structures found in nature. One of the most common shapes used by viruses is called icosahedral symmetry. An icosahedron is a geometric shape with 20 triangular faces, similar to certain dice used in board games. In viruses, the capsid proteins arrange themselves into this shape to create a strong and balanced protective shell around the genetic material. The structure is extremely efficient because it allows the virus to build a sturdy container using many identical protein pieces. To picture it, imagine a soccer ball made from triangular panels instead of pentagons and hexagons. The repeating triangles create a solid, symmetrical shell that can hold material inside. This design is very common in nature because it provides maximum internal space while using minimal building materials. For viruses with limited genetic instructions, this efficient geometric structure helps them build stable capsids without needing complex designs. The icosahedral shape used by many viruses is not just visually symmetrical, it also provides excellent structural stability. With 20 triangular faces connected together, the shape distributes pressure evenly across the entire capsid. You can think of this like a geodesic dome or a well-built soccer ball. These structures are strong because their triangular sections support each other, preventing weak points. For viruses, this design is extremely useful. They must protect delicate genetic material while traveling through environments that may include heat, chemicals, or physical stress. The icosahedral capsid creates a strong outer shell using very few types of proteins. This efficiency is important because viruses have very small genomes and cannot carry instructions for building complicated structures. By using simple repeating protein units arranged in a stable geometric pattern, viruses achieve maximum strength with minimal genetic instructions. For a long time, scientists believed viruses were always extremely tiny and simple. But the discovery of giant viruses changed that idea. Giant viruses are much larger than typical viruses and contain far more genes than scientists expected. Some of them are even large enough to be seen with microscopes that normally detect bacteria. To understand how unusual this is, imagine expecting something to be the size of a grain of sand, only to discover it's closer to the size of a pebble. That's roughly the surprise scientists experienced when giant viruses were discovered. Because of their large size and complex genetic content, these viruses blur the traditional boundary between viruses and living cells. Some giant viruses even carry genes that were once thought to exist only in cellular organisms. Their discovery forced scientists to rethink what viruses are and how they may have evolved. Most people imagine viruses as extremely tiny particles, far smaller than bacteria. While this is usually true, some giant viruses are actually larger than certain bacteria. Typical viruses are only about 20 to 300 nanometers wide, But giant viruses can reach hundreds of nanometers, or even over 1,000 nanometers in size. That means they can sometimes be bigger than small bacterial cells. To visualize the difference, imagine expecting something to be the size of a grain of dust, only to find it closer to the size of a small grain of sand. In the microscopic world, that's a huge jump. This discovery surprised scientists because size had long been one of the main ways viruses were distinguished from bacteria. Giant viruses show that the boundary between viruses and cellular life is not as clear as once believed, making them some of the most intriguing discoveries in modern microbiology. One of the first giant viruses ever discovered is called mimivirus. Scientists identified it in the early 2000s while studying microbes that infect amoebas, a type of single-celled organism. At first, researchers thought they were looking at a bacterium, not a virus. That's because mimivirus is unusually large for a virus, large enough to be seen under microscopes normally used to observe bacteria. The name mimivirus comes from the phrase mimicking microbe. It was named this way because it originally fooled scientists into thinking it was a small cellular organism rather than a virus. To picture the scale difference, imagine expecting something as tiny as a dust particle, only to find something closer to a small grain of sand under the microscope. The discovery of mimivirus forced scientists to reconsider long-held ideas about how small and simple viruses are supposed to be. Most viruses have very small genomes. Some carry fewer than 10 genes, just enough instructions to enter a cell and reproduce. Mimiviruses, however, are very different. They contain hundreds of genes, which is extremely unusual for a virus. To understand the scale, imagine comparing a tiny instruction pamphlet to a thick handbook. Typical viruses carry the pamphlet, while mimiviruses carry something much closer to the handbook. Many of these genes are involved in processes that scientists previously thought only living cells could perform. This made researchers question whether giant viruses might be more complex than traditional viruses. Despite having many genes, mimiviruses still depend on host cells to reproduce, which is why they are still classified as viruses. Their large genomes show that viruses can be far more complex than scientists once believed, expanding our understanding of the viral world. The discovery of giant viruses has made scientists rethink the traditional boundary between viruses and living cells. For a long time, viruses were considered extremely simple, tiny packages of genetic material with only a few genes. But giant viruses challenge that idea. Some of them carry hundreds or even over 1,000 genes, which is much more than typical viruses. Certain genes are involved in processes that were once believed to exist only in cellular life. You can imagine the difference, like comparing a small instruction note to a thick manual. The larger set of instructions makes giant viruses seem much more complex than scientists expected. Because of this, researchers sometimes debate where giant viruses truly belong in the tree of life. While they still rely on host cells to reproduce, their complexity makes them closer to cellular organisms than traditional viruses in some ways. Many giant viruses are known to infect amoebas, which are single-celled organisms, commonly found in water, soil, and moist environments. Amoebas are much larger than bacteria, making them suitable hosts for these unusually large viruses. Because amoebas are big cells, they provide more space and resources for giant viruses to replicate. This allows the viruses to carry larger genomes and produce many complex components inside the host. You can imagine an amoeba as a large workshop, while the virus is a machine that moves in and uses the workshop's tools to build copies of itself. Scientists often study giant viruses by observing infections in amoebas under microscopes. Amoebas naturally swallow particles from their environment, and sometimes they accidentally engulf giant viruses, which then begin the infection process. These interactions have helped researchers discover and study many giant viruses that were previously unknown. Because giant viruses are so large and complex, Some scientists believe they may represent very ancient forms of life. Their unusual genes and structures have raised questions about how viruses originally evolved. One idea is that giant viruses might be the remains of ancient cellular organisms that existed billions of years ago. Over time, these organisms may have lost many of their original functions and eventually became the viruses we see today. You can imagine this like an old machine that slowly lost parts over generations, leaving behind a simpler but still recognizable system. Another possibility is that giant viruses evolved by gradually collecting genes from different organisms they infected over millions of years. Scientists are still debating which explanation is correct. Regardless of their origin, giant viruses provide valuable clues about how life on Earth may have evolved in its earliest stages. Viruses have been discovered in some of the most extreme environments on Earth. Scientists have found them in places where conditions seem far too harsh for life. For example, viruses exist in boiling hot springs, deep ocean hydrothermal vents, freezing polar ice, and extremely salty lakes. These environments can reach temperatures hotter than boiling water or remain frozen for thousands of years. You can imagine these places like Earth's natural laboratories, where only the toughest organisms survive. Even in these extreme settings, viruses still manage to persist by infecting the microbes that live there. Many of the microbes that survive in extreme environments are called extremophiles, and viruses have evolved to infect them as well. This shows how adaptable viruses can be. Wherever life exists, even in the harshest conditions, viruses are usually there alongside it, interacting with the organisms that live in those environments. Scientists have discovered viruses in environments that seem completely opposite from each other, including scorching hot springs and frozen polar ice. These places represent some of the most extreme conditions on Earth. Hot springs can reach temperatures close to boiling water, sometimes above 90 degrees Celsius. Yet certain microbes thrive there, and viruses that infect those microbes exist alongside them. At the other extreme, viruses have also been found in glaciers and polar ice, where temperatures remain far below freezing. Some of these viral particles may remain trapped in ice for extremely long periods. To imagine this contrast, think of viruses surviving in places as different as a steaming hot kettle and a deep freezer. These discoveries show that viruses can exist anywhere their host organisms live, even in environments that appear too hot or too cold for most life. Scientists have discovered that some viruses can remain infectious even after being frozen in ice for thousands of years. In frozen environments like glaciers and permafrost, extremely low temperatures can preserve viral particles for very long periods. Ice works almost like a natural freezer, slowing down chemical reactions that would normally break down the virus. As long as the viral structure and genetic material stay intact, the virus can potentially become active again if it enters a suitable host. Researchers have successfully revived ancient viruses from permafrost samples that were tens of thousands of years old. Once thawed and introduced to host cells in laboratory conditions, these viruses were still able to infect their hosts. You can imagine this like a seed frozen in time, remaining dormant until the right conditions allow it to grow again. These discoveries show how long viruses can persist in extreme environments. Viruses can move through the air as tiny particles, allowing them to travel from one place to another. When infected people cough, sneeze, talk, or even breathe, microscopic droplets can be released into the air. Some of these droplets may contain virus particles. Many of these droplets are extremely small, far smaller than a grain of dust, so they can remain suspended in the air for a period of time before settling. This allows viruses to spread through the surrounding environment. You can imagine this like fine mist from a spray bottle. Once released, the tiny droplets drift through the air before eventually landing on surfaces or being inhaled by others. However, Not all viruses spread this way. Some viruses rely on other methods, such as insect bites or direct contact. Understanding how viruses travel through the air helps scientists develop better ways to reduce the spread of infections. Viruses released into the air don't always stay near where they originated. Air currents and wind can carry viral particles across surprisingly long distances. Tiny droplets, or particles containing viruses, can be lifted by moving air and transported through buildings, across cities, and sometimes even across large regions. Because these particles are extremely small, often much smaller than dust, they can remain suspended in the air for extended periods. You can imagine this like pollen drifting in the wind during spring. Just as pollen grains can travel far from the plant that released them, virus-containing particles can move through the air as well. In outdoor environments, wind patterns and weather conditions influence how far particles travel. Indoors, ventilation systems and airflow can also affect how viruses spread through the air. Understanding how air currents move particles helps scientists design better ventilation and infection control strategies. Many viruses spread through respiratory droplets that are released when people cough, sneeze, talk, or even laugh. These droplets are tiny particles of moisture that come from the nose and mouth. When someone coughs or sneezes, thousands of droplets can be launched into the air. Some are large and fall to the ground quickly, while others are small enough to float briefly before settling. You can picture this like a burst of fine mist from a spray bottle. The droplets spread outward and eventually land on nearby surfaces, or may be inhaled by someone close by. If the droplets contain virus particles and another person breathes them in, infection may occur. This is why covering your mouth when coughing and maintaining distance during illness can reduce transmission. These simple actions help limit how far infectious droplets travel through the air. Not all viruses spread directly through the air. Some can spread when people touch contaminated surfaces, a process often called surface transmission. When an infected person coughs, sneezes, or touches objects, tiny droplets containing viruses can land on surfaces like door handles, tables, phones, or keyboards. If another person touches that surface and then touches their eyes, nose, or mouth, the virus may enter the body. You can imagine this like invisible fingerprints left behind on objects. Even though the surface may look clean, microscopic virus particles can remain there for a period of time. However, viruses don't survive forever on surfaces. Their survival depends on factors such as temperature, humidity, and the type of material. This is one reason why regular cleaning and hand washing are effective. Soap and disinfectants can break apart virus particles, reducing the chance of infection. Not all viruses spread directly from person to person. Some viruses rely on insects to carry them between hosts. These insects act as vectors, meaning they transport the virus from one organism to another. Mosquitoes are one of the most well-known examples. When a mosquito bites an infected animal or person, it may pick up virus particles along with the blood. Later, when the mosquito bites another host, the virus can be passed along during the feeding process. You can think of the mosquito like a tiny flying syringe, moving microscopic virus particles between hosts without even knowing it. Other insects, such as ticks or certain flies, can also transmit viruses. This method of spread allows viruses to travel between animals and sometimes humans, even if the hosts never directly interact with each other. Vector transmission plays a major role in the spread of several important viral diseases. Mosquitoes are responsible for spreading several well-known viruses, including dengue and Zika. These viruses cannot move on their own from one host to another, so they rely on mosquitoes to act as carriers. When a mosquito bites an infected person, it can pick up virus particles from the blood. The virus then develops inside the mosquito for a short period. Later, when the mosquito bites another person, the virus can enter the new host through the bite. You can imagine the mosquito as a tiny flying delivery system, unknowingly transporting viruses between people. Because mosquitoes breed quickly and live in many parts of the world, they can spread viruses across large populations. Warm climates and standing water often allow mosquito populations to grow rapidly. This is why controlling mosquito populations, such as removing standing water, can help reduce the spread of certain viral diseases. Just like viruses that infect humans can spread through insect bites, some plant viruses are also transmitted by insects. Many of these viruses rely on insects, such as aphids, leafhoppers, or whiteflies, to move from one plant to another. When an insect feeds on a plant, it pierces the plant tissue with its mouthparts and drinks the plant's sap. If the plant is infected, the insect may pick up virus particles during feeding. Later, when the same insect feeds on a different plant, the virus can enter the new plant through the tiny feeding wound. You can imagine this process like a needle transferring tiny droplets between plants. This method allows plant viruses to spread quickly through fields and forests. In agriculture, this can sometimes lead to large crop infections, affecting plants like tomatoes, potatoes, and other important food crops. Certain viruses can spread through contaminated water, especially when water sources become polluted with virus particles from infected organisms. This can happen when sewage or waste enters rivers, lakes, or drinking water supplies. If people drink or use this contaminated water, virus particles may enter the body and cause infection. You can imagine this like adding a drop of dye into a glass of water. The color spreads throughout the water, even though the original drop was tiny. In a similar way, microscopic virus particles can disperse through water systems. Because viruses are so small, they can sometimes pass through water unnoticed unless proper treatment and filtration systems are used. For this reason, clean water systems and sanitation are extremely important for public health. Water treatment plants help remove or destroy viruses so that drinking water remains safe for human use. During a viral outbreak, viruses can evolve very quickly as they spread through a population. Every time a virus infects a new host and reproduces, small genetic changes, called mutations, may occur. Because viruses replicate so rapidly, these mutations can accumulate in a short period of time. In a large outbreak where millions of infections occur, the virus is essentially copying itself millions of times, creating many opportunities for new genetic variations. You can think of this like making millions of photocopies of a document. Occasionally, small copying errors appear, and some of those changes might affect how the virus behaves. Most mutations have little effect, but sometimes a change can make the virus spread more easily or behave differently. By studying these genetic changes, scientists can track how viruses evolve during outbreaks and better understand how they move through populations. Because some viruses evolve quickly, developing vaccines for them can sometimes be more challenging. When a virus changes its genetic makeup through mutations, certain parts of the virus may look slightly different to the immune system. Vaccines usually work by training the immune system to recognize specific viral structures. If those structures change too much over time, the immune system may not recognize the virus as effectively as before. You can imagine this like a lock that keeps slightly changing shape. A key that once fit perfectly might not work as well if the lock changes. However, it's important to understand that many vaccines still remain highly effective, even when viruses evolve. Scientists can also update vaccines when needed to match new viral versions. Tracking viral mutations helps researchers adjust vaccine strategies and continue protecting people against changing viral strains. Vaccines work by teaching the immune system how to recognize a virus before a real infection occurs. Instead of exposing the body to the full disease, vaccines introduce a harmless piece of the virus or a weakened version of it. This allows the immune system to study the virus and prepare defenses without causing serious illness. Special immune cells learn to recognize the virus and create protective antibodies that can attack it later. You can imagine this process like a training exercise for security guards. The guards study a picture of a potential intruder so they can quickly recognize and stop them if they appear in the future. Once trained, the immune system can often respond much faster and more effectively when the real virus enters the body. Because of this early preparation, vaccines help prevent infections or reduce how severe an illness becomes. The first successful vaccine in history was created to protect against smallpox, a deadly viral disease that once affected millions of people. In 1796, a doctor named Edward Jenner made an important observation. He noticed that milkmaids who had previously caught cowpox, a much milder disease from cows, rarely became infected with smallpox. To test this idea, Jenner introduced material from a cowpox sore into a young boy. Later, when the boy was exposed to smallpox, he did not become sick. The earlier exposure had trained his immune system to recognize and fight the virus. You can think of this like a practice drill for the immune system, preparing it for a future threat. Jenner's discovery led to the concept of vaccination. The word vaccine actually comes from vacca, the Latin word for cow, honoring the cowpox connection. Smallpox is one of the few diseases in human history to be completely eradicated worldwide. In 1980, the World Health Organization officially declared that the virus no longer existed in natural human populations. Before eradication, smallpox was one of the most feared diseases on Earth. It caused severe illness, painful skin lesions, and had a death rate of about 30% in many outbreaks. For centuries, it killed millions of people. The success of eradication came from a massive global vaccination campaign. Health workers traveled across countries and remote communities to vaccinate people and stop the virus from spreading. You can imagine this effort like closing every possible door the virus could use to move from person to person. Because smallpox only infected humans and had an effective vaccine, scientists were able to eliminate it completely. One of the greatest achievements in medical history. The elimination of smallpox is widely considered one of the greatest achievements in the history of medicine. For thousands of years, this virus caused devastating outbreaks across the world. Smallpox didn't just make people sick. It often left survivors with permanent scars, and many people lost their eyesight after infection. Entire communities could be affected during major outbreaks. What makes the eradication so remarkable is that humans completely stopped the virus from spreading anywhere on Earth. Through coordinated vaccination programs, surveillance, and rapid response to new cases, scientists and health workers gradually eliminated the virus from every region. You can imagine this effort like extinguishing a massive wildfire by putting out every remaining spark. Today, smallpox exists only in a few highly controlled laboratories for research purposes. Its eradication proved that, with enough global cooperation, humanity can defeat even deadly viral diseases. Viruses have played a major role in shaping human history, societies, and populations. Throughout history, viral outbreaks have influenced wars, migrations, economies, and even the rise and fall of civilizations. For example, when diseases spread through populations that had little or no immunity, they could cause massive loss of life in a short time. Entire communities sometimes experienced dramatic population declines after large epidemics. You can imagine a virus acting like an invisible force that changes the course of events, similar to how natural disasters can reshape societies. These outbreaks often pushed societies to improve medicine, sanitation, and public health systems. Many modern health practices, such as vaccination programs, disease monitoring, and quarantine measures, developed partly because of viral epidemics. By studying past viral outbreaks, scientists and historians can better understand how diseases influenced human development and global history. Influenza, often called the flu, is caused by a virus that has triggered several major pandemics in human history. A pandemic occurs when a disease spreads across many countries or continents and affects large numbers of people. Some influenza pandemics have killed millions of people worldwide. These events usually happen when a new strain of the virus appears, one that human immune systems have little experience fighting. You can think of this like a completely new disguise that the immune system doesn't recognize right away. Because the body has not seen the virus before, it may take longer to respond effectively. Influenza spreads easily through respiratory droplets when infected people cough, sneeze, or talk. Since people travel and interact frequently, the virus can move quickly through populations. These historical pandemics helped scientists understand how quickly viruses can spread and why global monitoring of viral diseases is so important. The influenza pandemic of 1918, often called the Spanish flu, is one of the most severe disease outbreaks in recorded history. Scientists estimate that it infected about 1/3 of the world's population at the time. To picture the scale, imagine 1 out of every 3 people on Earth becoming sick during the same global event. In a world population of about 1.8 billion people back then, that meant roughly 500 million infections. The virus spread rapidly across continents, partly because it occurred during the final months of World War I, when large numbers of soldiers were traveling between countries. Unlike many flu outbreaks, this pandemic affected not only the elderly and very young, but also many healthy young adults. In total, the pandemic is estimated to have caused tens of millions of deaths worldwide. It remains a powerful reminder of how quickly viruses can spread through global populations. Viruses do not only affect humans, they can also have major impacts on animal populations, sometimes spreading rapidly through entire species. When a virus infects animals that have little immunity, it can move through the population quickly, especially if the animals live in close groups. In some cases, large numbers of animals may become sick at the same time. You can imagine this like a wildfire moving through a dry forest, where the infection spreads rapidly from one animal to another. These outbreaks can affect wildlife, livestock, and marine animals. In certain situations, viral diseases have caused large declines in animal populations in specific regions. Such events can also influence ecosystems. When a large number of animals are affected, it may change food chains, predator-prey relationships, and environmental balance. This is why scientists monitor viruses not only in humans, but also in wildlife and animal populations. In some cases, viral infections can cause massive die-offs in wildlife populations. When a highly contagious virus spreads through animals that have little immunity, large numbers may become infected in a short time. These events sometimes occur in fish, birds, marine mammals, or land animals. Because wildlife populations often live in close groups or migrate together, viruses can spread quickly through entire communities. You can imagine this like a domino effect. Once the virus infects a few animals, it can move rapidly from one individual to the next. Such outbreaks can temporarily reduce animal populations and affect ecosystems. For example, if a large number of animals disappear, predators may lose a food source, or certain species may suddenly become more common. Scientists carefully study these die-offs because they help reveal how viruses move through natural ecosystems and animal populations. Viruses don't just affect humans and animals. They can also infect plants used in agriculture. Certain plant viruses can spread through fields and damage important crops, such as tomatoes, potatoes, wheat, and rice. When plants become infected, they may develop discolored leaves, stunted growth, or reduced fruit production. In severe cases, entire harvests can be affected, which can impact food supplies and farmers' livelihoods. You can imagine this like a silent problem spreading through a garden, where plants that once grew healthy suddenly begin producing less or showing unusual patterns on their leaves. Many plant viruses are spread by insects that feed on plant sap, allowing the virus to move from one plant to another. Because agriculture depends heavily on healthy crops, scientists study plant viruses carefully. Understanding how they spread helps farmers develop better ways to protect crops and maintain food production. Plant viruses can sometimes cause severe damage to crops, especially when they spread quickly through large farming areas. Unlike animals, plants cannot move away from infection, which means a virus can spread steadily across fields. When crops become infected, plants may grow more slowly, produce fewer fruits, or develop distorted leaves and unusual color patterns. These symptoms can reduce both the quantity and quality of the harvest. To picture the impact, imagine a large farm where thousands of plants suddenly stop producing properly. Even a small reduction in yield across a big field can lead to significant losses for farmers. Because agriculture feeds billions of people, plant viral diseases are taken very seriously. Farmers and scientists work together to control them by using resistant plant varieties, managing insect carriers, and monitoring crop health. Understanding plant viruses helps protect global food supplies and agricultural economies. Viruses may be simple, but they have played an important role in helping scientists understand how genetics works. Because viruses carry genetic material and interact closely with host cells, they became valuable tools for studying how genes function. In many experiments, scientists used viruses to observe how genetic instructions move, change, and control biological processes inside cells. Since viruses often reproduce quickly, researchers could study genetic changes much faster than in larger organisms. You can think of viruses as tiny biological test systems. Their simplicity makes it easier for scientists to observe how genetic information behaves. Some of the earliest discoveries about how genes are copied and expressed were made using viruses that infect bacteria. These experiments helped reveal basic rules of molecular biology. Even today, viruses remain important tools for researchers studying genetics, evolution, and how cells operate at the molecular level. Viruses played a major role in helping scientists understand how DNA carries genetic information. In the mid-20th century, researchers used viruses that infect bacteria, called bacteriophages, to study how genetic instructions control living processes. One famous set of experiments used these viruses to determine whether DNA or proteins carried genetic information. By carefully labeling viral DNA and proteins and observing how the virus infected bacteria, scientists discovered that DNA was the molecule responsible for passing genetic instructions. You can imagine this experiment like tracking two different colored packages to see which one enters a building. The results clearly showed that DNA entered the cell and directed the infection. This discovery was a turning point in biology. It helped scientists understand how genes work and eventually contributed to breakthroughs such as genetic engineering, biotechnology, and modern medicine. Some viruses contain special proteins called enzymes that scientists use in biotechnology and research. Enzymes are molecules that help speed up chemical reactions inside cells. Certain viruses bring their own enzymes because they need them to copy their genetic material or interact with host cells. Researchers discovered that these viral enzymes can also be very useful tools in laboratories. You can imagine these enzymes like specialized tools in a toolbox, each designed for a particular job. Scientists can isolate these tools and use them to study DNA, copy genetic material, or analyze biological samples. For example, some viral enzymes help researchers copy or modify genetic sequences in experiments. These tools have become important in fields like genetic engineering, medical diagnostics, and biotechnology. Even though viruses are often associated with disease, their enzymes have helped scientists develop powerful technologies for studying life at the molecular level. Scientists have discovered that viruses can be turned into helpful tools called viral vectors for gene therapy. In this approach, researchers modify viruses so they can deliver useful genes into human cells without causing disease. Normally, viruses are very good at entering cells and inserting their genetic material. Scientists take advantage of this natural ability by removing the harmful parts of the virus and replacing them with healthy genetic instructions. You can think of a viral vector like a delivery truck carrying a replacement part. Instead of spreading infection, the virus delivers a corrected gene to cells that need it. This technique is being used to treat certain genetic conditions, where a person's cells are missing a working gene. By adding the correct instructions, scientists hope to help the cells function properly again. Gene therapy using viral vectors is one of the most exciting areas of modern medical research. Viral vectors are especially useful because viruses are naturally very good at entering human cells and delivering genetic material. Scientists take advantage of this ability when designing gene therapies. In these treatments, the virus is carefully modified so it cannot cause disease. Researchers remove the harmful viral genes and insert a healthy gene that doctors want to deliver into the patient's cells. You can imagine the process like sending a package through a highly reliable delivery service. The viral vector carries the genetic instructions and deposits them inside the target cell. Once the new gene is delivered, the cell may begin producing the missing or corrected protein needed for normal function. This technique is being studied for several genetic conditions and certain diseases. By using viruses as delivery vehicles, scientists can reach cells in ways that would otherwise be very difficult with traditional medicine. Scientists are exploring ways to use viruses as tools for fighting cancer. Instead of causing disease, certain viruses can be engineered or selected to infect and destroy cancer cells. These viruses are designed to target tumor cells more than healthy cells. Once the virus enters a cancer cell, it begins replicating inside it. Eventually, the infected cancer cell bursts, releasing new virus particles that can move on to attack nearby cancer cells. You can imagine this like sending tiny biological agents into a tumor, where they multiply and gradually break apart the harmful cells. Another benefit is that when cancer cells are destroyed, the immune system may become more aware of the tumor, helping the body recognize and attack remaining cancer cells. Although this approach is still being studied and refined, virus-based therapies are showing promise as a new strategy in cancer treatment. Scientists have developed special viruses that are engineered to selectively infect and destroy cancer cells, while leaving most healthy cells unharmed. These modified viruses are designed to recognize features that are common in tumor cells. Cancer cells often grow rapidly and have certain weaknesses in their defense systems. Engineers design viruses to take advantage of these weaknesses, allowing the virus to replicate mainly inside cancer cells. You can picture this like a guided missile programmed to lock onto a specific target. The virus enters the tumor cell, multiplies inside it, and eventually causes the cell to burst. When the cancer cell breaks apart, it releases new virus particles that can move on to infect other nearby cancer cells. At the same time, the destruction of tumor cells can alert the immune system, helping it recognize and attack the cancer more effectively. Viruses that are designed to attack and destroy cancer cells are known as oncolytic viruses. The word oncolytic comes from terms meaning tumor breaking. These viruses are either naturally able to infect cancer cells or are genetically modified by scientists to specifically target tumors. Once they enter a cancer cell, the virus begins replicating inside it. Eventually, the infected cancer cell bursts open, releasing many new virus particles. These new viruses can then move on to infect nearby cancer cells, continuing the cycle. You can imagine this like a chain reaction inside a tumor, where each destroyed cancer cell releases more virus particles that attack the next one. Another benefit is that the destruction of cancer cells can alert the immune system. The immune system may begin recognizing the tumor as a threat and help attack remaining cancer cells. Researchers are increasingly studying viruses as tools for new medical treatments. Although viruses are often associated with disease, their natural ability to enter cells and deliver genetic material makes them useful for many medical applications. Scientists are exploring ways to use viruses in gene therapy, cancer treatments, and vaccine development. In these cases, viruses are carefully modified so they cannot cause illness, but can still perform helpful tasks inside the body. You can imagine these modified viruses as tiny delivery systems, carrying helpful genetic instructions or targeting harmful cells. Because viruses interact so closely with cells, they can reach places that many traditional medicines cannot easily access. This gives scientists new possibilities for treating certain diseases. As research continues, viruses may become important tools in future medicine, helping doctors develop more precise and targeted therapies. Viruses and their hosts often evolve side by side over long periods of time. As hosts develop defenses against viruses, the viruses may also change and adapt in response. This creates a constant cycle of adaptation, sometimes described as an evolutionary arms race. The host's immune system develops ways to recognize and block viruses, while the viruses evolve new strategies to enter cells or avoid detection. You can imagine this like two teams constantly adjusting their strategies in a long game. Each side learns from the other and changes over time. Because viruses reproduce so quickly, they can evolve rapidly. Hosts, on the other hand, evolve more slowly over generations. This ongoing interaction has influenced the evolution of many species. Over millions of years, viruses and their hosts have shaped each other's biology, leaving traces of their interactions in genetic material. Some viruses have existed alongside certain species for millions of years, evolving together over extremely long periods of time. Instead of constantly causing severe disease, many of these viruses have developed a kind of balance with their hosts. In these cases, the host's immune system and the virus gradually adapt to each other. The virus may become less harmful, while the host develops defenses that keep the infection under control. You can imagine this like two neighbors learning to live side by side. At first, there may be conflict, but over time a stable relationship forms. Scientists have found evidence of long-term virus-host relationships by studying genetic material and viral fragments inside genomes. This long coexistence shows that viruses are not always temporary visitors. In some cases, they have been interacting with certain species since ancient times, influencing their biology and evolution. Just like fossils preserved in rock tell us about ancient animals, scientists have discovered viral fossils hidden inside DNA. These genetic traces are pieces of viral material that became permanently inserted into the genomes of organisms. Long ago, over millions of years, viruses infected ancient species and sometimes inserted their genetic material into reproductive cells. When this happened, the viral DNA could be passed down from generation to generation. You can imagine this like an old note accidentally left inside a book and copied every time the book is printed again. Even if the note is no longer useful, it remains part of the book. By studying these viral fossils, scientists can learn about viruses that existed millions of years ago. Even if those viruses no longer exist today, these genetic clues allow researchers to reconstruct parts of ancient viral history and evolution. Scientists can learn about ancient viruses by studying the genomes of living organisms. A genome is the complete set of genetic instructions inside a cell. Hidden within these genomes are fragments of viral DNA, left behind by infections that happened millions of years ago. These genetic fragments act like historical records, allowing researchers to trace how viruses evolved and interacted with their hosts over long periods of time. You can imagine this process like reading layers in a history book, where each layer reveals events that happened long before humans were around. By comparing viral sequences found in different species, scientists can estimate when certain viruses appeared and how they spread between organisms. This helps researchers build evolutionary timelines, showing how viruses changed through history. Studying viral history through genomes gives scientists valuable clues about how viruses evolved and how they may behave in the future. Some scientists believe that viruses may have existed since the earliest stages of life on Earth, possibly billions of years ago. Although we cannot see ancient viruses directly, clues from genetics suggest that viruses have been interacting with living organisms for an extremely long time. Life on Earth is thought to have appeared around 3.5 to 4 billion years ago, and viruses may have emerged not long after the first cells developed. You can imagine the earliest ecosystem on Earth as a microscopic world filled with primitive cells and tiny genetic particles interacting with them. Viruses could have been part of that ancient biological environment from the very beginning. Because viruses depend on cells to reproduce, many scientists think they evolved alongside the first life forms. Their long history means viruses may have influenced the evolution of life for billions of years. Despite decades of research, scientists still do not fully agree on where viruses originally came from. The origin of viruses remains one of the most interesting mysteries in biology. One reason it's difficult to answer is that viruses do not leave traditional fossils like bones or shells. Because they are made mostly of genetic material and protein, they rarely leave physical traces in ancient rocks. Instead, scientists must study genetic clues found in modern organisms and viral genomes. These clues provide hints about how viruses may have evolved long ago. You can think of this like trying to solve an ancient puzzle, using only scattered pieces of evidence. Several theories exist about how viruses began, and each one explains certain features of viruses but not all of them. Because of this, researchers continue studying viral genetics and evolution to better understand their mysterious origins. One scientific theory suggests that viruses may have evolved from pieces of genetic material that escaped from living cells long ago. According to this idea, small fragments of DNA or RNA somehow gained the ability to move between cells and copy themselves. You can imagine this like pages from a manual breaking free and starting to travel between different machines. Over time, these genetic fragments may have developed protective protein shells and mechanisms for entering new cells. As these fragments continued evolving, they eventually became the viruses we recognize today. This idea is sometimes called the escape hypothesis. Supporters of this theory point out that some viruses contain genes that look very similar to genes found in cellular organisms. This suggests that the viruses may have originally borrowed or inherited genetic material from cells. While this theory explains certain features of viruses, scientists are still studying whether it fully explains how viruses first appeared. Another scientific theory suggests that viruses may have evolved from ancient cellular organisms that gradually became simpler over time. According to this idea, some early life forms might have once been full cells with many biological functions. Over millions of years, these organisms may have lost many of their internal structures and abilities, eventually becoming the much simpler viruses we see today. You can imagine this like an old machine that slowly loses parts until only the essential components remain. The remaining system still works in a limited way, but now depends on other machines to function. Supporters of this theory point out that some giant viruses carry large genomes and complex genes, which might be leftovers from more complex ancestors. Although this explanation fits some observations, scientists are still debating whether viruses truly evolved from ancient cells or followed a different evolutionary path. A third theory proposes something even more surprising. Viruses might have existed before the first true cells appeared on Earth. According to this idea, early Earth may have contained simple, self-replicating genetic molecules long before complex cellular life developed. In this ancient world, small strands of genetic material may have been able to copy themselves and interact with other molecules in the environment. Over time, some of these primitive genetic systems might have evolved into viruses. You can imagine this like early prototypes appearing before full machines were built. These simple systems may have existed before the first cellular machines of life were fully developed. If this theory is correct, viruses could represent remnants of a very ancient stage of life's history. However, proving this idea is difficult because such early biological systems existed billions of years ago, leaving very little direct evidence behind. Even today, scientists continue to debate how viruses originally appeared on Earth. Several different theories exist, and each one explains certain aspects of viral biology, but none of them fully answers every question. The three main ideas suggest that viruses may have come from escaped genetic material, ancient cellular organisms, or primitive genetic systems that existed before cells. Each theory has supporting evidence, but scientists are still collecting more data to understand which explanation fits best. You can imagine this situation like trying to reconstruct a very old story using only fragments of the original pages. Some parts of the story are clear, while others remain uncertain. New discoveries, especially involving giant viruses and genetic analysis, continue to provide fresh clues. As research advances, scientists hope to better understand how viruses began and how they have shaped life on Earth over billions of years. It may sound strange, but some viruses can actually infect other viruses. These unusual viruses are known as virophages. A virophage cannot infect cells on its own. Instead, it takes advantage of a situation where a giant virus is already infecting a host cell. The virophage enters the same cell and interferes with the giant virus while it is reproducing. You can imagine this like a tiny parasite attacking a much larger parasite. The giant virus is busy using the cell to make copies of itself, and the virophage sneaks in and disrupts the process. As the virophage replicates, it can reduce the number of giant virus particles that are produced. This surprising interaction shows that the viral world is more complex than scientists once believed, with viruses sometimes interacting with and affecting each other in unexpected ways. Viruses that infect other viruses are known as virophages. The name combines the word virus with the Greek word phage, meaning eater. Although they don't literally eat viruses, they interfere with the way certain viruses reproduce. Virophages typically appear when a giant virus is already infecting a host cell. The giant virus begins using the cell's machinery to make new virus particles. At the same time, the virophage enters the same cell and starts using the viral replication system for its own reproduction. You can imagine this situation like a stowaway sneaking onto a factory assembly line. The giant virus sets up the production line, and the virophage secretly uses that system to make copies of itself. In doing so, the virophage often disrupts the giant virus's ability to reproduce effectively, reducing the number of giant viruses produced. Virophages take advantage of giant viruses by hijacking the viral replication process during infection. When a giant virus infects a host cell, it creates special areas inside the cell called viral factories. Where new virus particles are assembled, the virophage enters the same cell and begins using this factory for its own reproduction. Instead of building only giant viruses, the system now starts producing virophage particles as well. You can imagine this like someone secretly redirecting a factory assembly line to produce a different product. The original factory was designed to build giant viruses, but the virophage changes the output. As the virophage multiplies, it often interferes with the giant virus's production process. This can reduce the number of giant virus particles that are created. In some cases, the presence of virophages may even protect host organisms by weakening the giant virus infection. Some viruses have the ability to hide inside bacteria for long periods of time without immediately destroying them. These viruses infect bacterial cells and then remain quietly inside instead of immediately producing new virus particles. In this hidden state, the virus's genetic material becomes part of the bacterium's DNA. The bacterium continues living and dividing normally, copying the viral DNA along with its own genetic instructions. You can imagine this like a hidden passenger traveling inside a train. The train continues moving and carrying passengers, while the hidden traveler quietly remains on board. As the bacteria multiply, the viral DNA spreads to new bacterial cells as well. At certain times, environmental changes or stress can trigger the virus to become active again. When that happens, the virus begins producing new virus particles and may eventually destroy the bacterial cell. When a virus hides inside a bacterium without immediately destroying it, the state is called lysogeny. In this stage, the virus inserts its genetic material into the bacterium's DNA and becomes part of the cell's genome. The viral DNA in this form is known as a prophage. As the bacterium grows and divides, it copies the viral DNA along with its own genetic instructions. This means the hidden virus can quietly spread to many new bacterial cells without producing virus particles right away. You can imagine this like a secret instruction page inserted into a recipe book. Every time the book is copied, the hidden page is copied as well. For long periods, the bacterium may continue living normally, but certain conditions, such as stress or environmental changes, can trigger the virus to leave this hidden state and begin actively producing new viruses. Viruses that are dormant inside cells do not always remain inactive forever. Certain environmental changes or stresses can trigger them to become active again. For example, changes such as radiation, chemical exposure, nutrient shortages, or sudden temperature shifts can disturb the host cell. When this happens, the virus may detect the change and switch from a dormant state to an active one. You can imagine this like an alarm system suddenly turning on when conditions change. The virus senses that something in the environment is different and begins producing new virus particles. Once activated, the virus starts copying its genetic material and assembling new viruses inside the cell. Eventually, the host cell may burst open, releasing the newly formed viruses. This ability to remain hidden and activate later helps viruses survive difficult conditions and spread when opportunities arise. Viruses can spread very quickly through populations, especially when people or animals live close together. Once a virus enters a group where individuals have little immunity, it can move from host to host in a short amount of time. Every infected person or animal may produce thousands or even millions of virus particles, each capable of infecting someone else. This rapid multiplication allows the infection to grow quickly. You can imagine this like a chain reaction of falling dominoes. One infection leads to several more, and those infections lead to even more. The speed of viral spread depends on several factors, such as how easily the virus moves between hosts, how often individuals interact, and how strong the immune defenses are in the population. Because viruses can spread so efficiently, public health measures like vaccination, monitoring, and early detection are important for controlling outbreaks. Modern global travel allows viruses to move between continents much faster than in the past. In earlier centuries, diseases spread mainly through slow travel routes, such as ships or long land journeys. Today, airplanes can carry passengers across the world in less than a day. If a person is infected but does not yet show symptoms, they may unknowingly travel long distances before realizing they are sick. You can imagine this like a spark being carried by the wind from one forest to another. The virus may start in one location but quickly appear thousands of kilometers away. Because millions of people travel internationally every day, viruses can reach new populations very quickly. For this reason, scientists and health organizations monitor outbreaks carefully and share information globally. Rapid communication and surveillance help detect and respond to viral threats before they spread widely. Monitoring viruses is an important part of protecting public health. Scientists and health organizations constantly watch for new viral infections, changes in existing viruses, and unusual outbreaks. This monitoring helps experts detect potential problems before they spread widely. For example, laboratories around the world collect samples, analyze viral genetic material, and track where infections are appearing. You can imagine this system like a global early warning network. Instead of watching for storms, scientists are watching for changes in viruses that might affect human or animal populations. By detecting new viruses or mutations early, health officials can respond faster with vaccines, treatments, travel advisories, or containment measures. This global cooperation is essential because viruses do not respect borders. Monitoring them helps scientists understand how viruses spread and allows countries to prepare for potential outbreaks before they become larger crises. Scientists around the world continuously track new viral strains to understand how viruses are changing over time. A strain is a slightly different version of a virus that has developed through mutations. As viruses replicate, small genetic changes can occur. Most of these changes have little effect, but occasionally they may influence how the virus spreads, how severe the infection becomes, or how well treatments and vaccines work. You can imagine this like watching different branches grow from the same tree. Each branch comes from the same original trunk, but it develops its own small differences. To monitor these changes, researchers collect virus samples from infected individuals and analyze their genetic sequences in laboratories. By comparing these sequences, scientists can map how viral strains evolve and spread across regions. This information helps public health experts adapt vaccines, treatments, and prevention strategies when needed. Viruses carry genetic instructions just like living organisms, but their genomes can be made of either DNA or RNA. A genome is the complete set of genetic information that tells the virus how to reproduce and function. Most living cells on Earth use DNA to store their genetic information. Viruses, however, are more flexible. Some viruses use DNA, while others use RNA as their genetic material. You can imagine this like two different types of instruction formats. Both formats contain information, but they are written in slightly different ways. This difference affects how the virus replicates inside host cells. RNA viruses often copy their genetic material quickly, which can lead to more frequent mutations. DNA viruses usually copy their genomes more carefully and tend to change more slowly. Understanding whether a virus uses DNA or RNA helps scientists determine how it behaves and how to control it. The genetic material inside a virus can come in different forms. Some viruses have single-stranded genomes, meaning their DNA or RNA consists of a single chain of genetic information instead of the usual double structure. Most people are familiar with DNA as a double helix, which looks like a twisted ladder with two strands. However, in single-stranded viruses, the genetic material is more like one side of that ladder instead of the full structure. You can imagine it like a zipper with only one side present. Even though only one strand exists, the virus can still use it to produce the instructions needed to replicate. Single-stranded genomes are common in many RNA viruses and some DNA viruses. When these viruses enter a host cell, the cell's machinery often creates a complementary strand, so the virus can copy its genetic material and produce new virus particles. While some viruses carry a single strand of genetic material, others have double-stranded genomes, meaning their DNA or RNA consists of two complementary strands paired together. This structure is similar to the double helix shape of human DNA, where two strands twist around each other like a spiral ladder. The paired strands help protect the genetic information and make it easier for the virus to copy its genome during replication. You can picture this like a two-sided zipper, where each side matches the other perfectly. If one side is damaged, the other strand can help guide the repair or copying process. Double-stranded genomes are common in many DNA viruses. Because the structure is more stable, these viruses often mutate more slowly than many RNA viruses. This stability can influence how the virus evolves and how scientists design treatments or vaccines to control infections. Not all viruses store their genetic information in one continuous piece. Some viruses have segmented genomes, meaning their genetic instructions are divided into several separate pieces. You can imagine this like a book that is split into multiple small chapters instead of one long scroll. Each segment contains part of the instructions needed for the virus to function and reproduce. When the virus infects a host cell, all of these segments work together to guide the production of new virus particles. However, the segmented structure also allows something interesting to happen. If two related viruses infect the same cell at the same time, their genetic segments can sometimes mix and combine. This can produce new combinations of genetic material in the newly formed viruses. Because of this mixing ability, viruses with segmented genomes can sometimes evolve and create new variants more easily. Viruses with segmented genomes have a special ability. They can mix genetic material between different viral strains. This process can occur when two related viruses infect the same host cell at the same time. Inside the infected cell, each virus releases its genetic segments. When new virus particles are assembled, the segments can sometimes combine in new ways, creating a virus with a mixture of genetic instructions from both original viruses. You can imagine this like shuffling two decks of cards together. When the cards are reshuffled, the new deck contains a different combination than either of the original decks. This mixing process is known as genetic reassortment. It can sometimes produce viruses with new characteristics, such as changes in how they spread or interact with the immune system. Because of this ability, viruses with segmented genomes can evolve and produce new variants relatively quickly. When viruses exchange genetic segments through reassortment, the result can be the creation of new viral variants. These variants contain combinations of genes that did not previously exist together. This happens when two related viruses infect the same host cell, and their genetic segments are reshuffled during the assembly of new virus particles. The newly formed viruses may carry some segments from one virus and others from the second virus. You can imagine this like mixing pieces from two different puzzles to create a new picture. The final image may look different from either of the original puzzles. Sometimes these new combinations make little difference, but occasionally a variant may spread more easily or interact differently with immune defenses. Because of this possibility, scientists carefully monitor viruses capable of genetic mixing to understand how new viral strains may appear over time. Viruses cannot produce their own energy. Unlike living cells, they do not have the machinery needed to generate energy from nutrients. Instead, viruses rely completely on the metabolism of the host cell. Metabolism is the system of chemical reactions that cells use to create energy and build molecules. When a virus infects a cell, it takes advantage of this system to power the production of new virus particles. You can imagine this like a machine that plugs into another machine's power supply. The virus doesn't have its own power source, so it uses the host cell's energy to operate. Because viruses depend so heavily on host cells, they cannot grow or reproduce independently, like bacteria or other organisms. This reliance is one reason scientists sometimes describe viruses as being on the edge between living and non-living, since they need living cells to become active. When viruses are outside a host cell, they are essentially inactive particles. They do not move on their own, produce energy, or carry out any biological processes. In this state, a virus is more like a sealed package of genetic instructions, waiting for the right opportunity. Its DNA or RNA is safely enclosed inside a protective protein shell, but nothing happens until it enters a living cell. You can imagine this like a program stored on a flash drive. The program exists, but it cannot run until the flash drive is plugged into a computer. Once a virus enters a suitable host cell, everything changes. The cell's machinery begins reading the viral instructions and producing new virus components. This ability to remain inactive outside cells helps viruses survive in the environment until they encounter a host where they can become active and begin replicating. When viruses are outside a host organism, they behave almost like inert particles rather than active living things. In this state, they do not grow, reproduce, or carry out chemical reactions. A virus outside a cell is simply a tiny package of genetic material wrapped in a protective shell. Without access to a host cell's machinery and energy supply, the virus cannot do anything on its own. You can picture it like a set of instructions sealed inside an envelope. The instructions are there, but nothing happens until someone opens the envelope and follows them. Because of this inactive state, viruses can sometimes remain on surfaces or in the environment for a period of time without changing. However, once a virus enters a suitable host cell, it can quickly switch from this inactive form into an active infection process. Even though viruses remain inactive outside a host, they can become active very quickly once they enter a suitable cell. As soon as the virus successfully attaches to a cell and releases its genetic material, the infection process begins. The cell's machinery immediately starts reading the viral instructions and begins producing viral proteins and genetic copies. This rapid activation allows the virus to take control of the cell in a short amount of time. You can imagine this like pressing the power button on a machine that has been sitting idle. The moment the power is turned on, the machine starts running. Because viruses are already fully assembled and ready to deliver their genetic instructions, the transition from inactive particle to active infection can happen very quickly after entry into a cell. This rapid activation is one reason viral infections can begin spreading soon after exposure. In today's interconnected world, viruses can spread between continents in just a few days. This rapid movement is largely due to modern transportation, especially international air travel. Millions of people fly between countries every day. If someone becomes infected with a virus before showing symptoms, they might travel long distances without realizing they are carrying the infection. You can imagine this like a spark being carried across oceans by fast-moving winds, starting new fires in distant places. In the same way, a virus that begins in one region can quickly appear thousands of kilometers away. Because airplanes can cross continents in less than 24 hours, viruses may reach new populations long before health officials detect the first cases. This is why global cooperation, early detection systems, and disease monitoring are so important. They help scientists identify and respond to viral threats before they spread widely. To protect public health, scientists around the world maintain systems for global virus surveillance. These systems monitor infections in humans, animals, and the environment to detect new viruses or unusual outbreaks as early as possible. Hospitals, research laboratories, and public health agencies regularly collect and analyze virus samples. Scientists study their genetic material to identify new strains or previously unknown viruses. You can think of this like a worldwide weather monitoring network, but instead of tracking storms, scientists are watching for signs of viral activity. When a new virus is detected, researchers quickly share information with international health organizations. This allows experts to study the virus, track how it spreads, and develop strategies such as vaccines, treatments, or containment measures. Early detection is crucial because responding quickly can help prevent small outbreaks from becoming global health emergencies. Some viruses change so quickly that their mutations can outpace how fast vaccines are updated. Because viruses replicate in huge numbers, small copying errors can happen frequently in their genetic material. When millions of virus particles are produced, these small changes can accumulate and create new viral variants. In some cases, these variants may look slightly different to the immune system compared to earlier versions of the virus. You can imagine this like a disguise that keeps changing over time. If the disguise changes too often, it may take time for defenses to recognize the new version. This is why scientists continuously monitor viral mutations and update certain vaccines when necessary. Even when viruses change, vaccines often still provide important protection against severe illness. Understanding how quickly viruses evolve helps researchers design better strategies to adapt vaccines and treatments over time. While some viruses change rapidly, others remain genetically stable for many years or even decades. This means their genetic material does not mutate very quickly, so the virus stays mostly the same over long periods of time. This stability often happens when a virus has a more reliable copying system for its genetic material, which reduces the number of errors during replication. As a result, fewer mutations occur. You can imagine this like a carefully copied document that rarely develops typos, even after being duplicated many times. Because these viruses change slowly, vaccines and treatments developed for them can remain effective for long periods. Scientists do not need to update vaccines as frequently when the virus remains stable. Understanding which viruses mutate quickly and which remain stable helps researchers plan long-term strategies for disease prevention and control. Viral outbreaks can affect far more than just health. They can also influence societies, economies, and daily life. When a virus spreads widely through a population, communities may change how they work, travel, and interact. For example, large outbreaks can lead to temporary closures of businesses, schools, and public events. Travel patterns may change, and healthcare systems may become extremely busy as doctors treat many patients at once. You can imagine this like a sudden storm disrupting normal activities across an entire city. Even though the storm eventually passes, it changes how people plan and prepare for the future. Throughout history, viral outbreaks have influenced government decisions, medical research, and public health policies. Many modern health systems and disease monitoring programs developed partly because societies needed better ways to respond to viral threats. These events show how viruses can shape human behavior and global systems. Over the past century, advances in medicine have greatly improved our ability to detect, prevent, and treat viral diseases. Scientists have developed vaccines, antiviral drugs, and advanced diagnostic tools that help reduce the impact of many viruses. Vaccination programs are one of the most powerful tools. By training the immune system to recognize viruses in advance, vaccines can prevent infections or make illnesses much less severe. You can imagine this like installing protective barriers before a storm arrives. Instead of reacting after damage occurs, prevention helps reduce the impact. Modern laboratories can also identify viruses quickly using genetic testing. This allows doctors to detect infections earlier and respond more effectively. Although viruses will always exist in nature, ongoing research and medical innovation continue to improve humanity's ability to control outbreaks and protect public health. In addition to vaccines, scientists have developed antiviral drugs that help treat certain viral infections. Unlike antibiotics, which kill bacteria, antiviral drugs are designed to interfere with specific stages of the viral life cycle. Many antiviral medications work by blocking the virus from copying its genetic material or assembling new virus particles inside the host cell. Without the ability to reproduce efficiently, the virus has a much harder time spreading through the body. You can imagine this like jamming the gears inside a machine. The machine might still exist, but it cannot operate properly once the key parts are blocked. Because viruses rely heavily on host cells, designing antiviral drugs can be challenging. Scientists must find ways to stop the virus without harming the body's own cells. Despite this challenge, antiviral treatments have become important tools for managing certain viral diseases. Viruses have helped scientists learn a great deal about how the immune system works. When a virus enters the body, the immune system must quickly recognize it as a threat and respond to stop the infection. By studying how the body reacts to viruses, researchers have discovered how immune cells identify foreign particles, produce antibodies, and remember past infections. You can imagine this like a security system, learning to recognize intruders. Once the system has seen a particular intruder before, it can respond faster the next time. Scientists often observe viral infections in laboratories to understand which immune responses are most effective. This knowledge helps researchers design better vaccines and treatments. In many ways, Viruses act like natural experiments that reveal how the body's defenses operate. Studying these interactions continues to improve our understanding of human immunity and disease protection. Viruses are extremely simple compared to living cells, yet they have an enormous impact on life on Earth. A virus may contain only a small amount of genetic material and a protein shell, but its influence can extend across ecosystems, species, and even evolutionary history, because viruses infect organisms ranging from bacteria to plants and animals. They affect countless biological processes. In the oceans, viruses help control microbial populations and recycle nutrients that support marine food webs. In medicine, viruses have shaped the development of vaccines, antiviral drugs, and modern genetic research. They have also influenced evolution by transferring genes and interacting with organisms over millions of years. You can imagine viruses as tiny forces with global influence. Even though they are microscopic and simple in structure, their presence has helped shape the biological world in powerful ways. Even after more than a century of research, viruses remain some of the most mysterious entities in biology. They are incredibly simple in structure, often just genetic material wrapped in a protein shell, yet they have enormous influence over life on Earth. Viruses exist in astonishing numbers, inhabit nearly every environment, and interact with almost every form of life. They shape ecosystems, drive evolution, influence global health, and even leave traces of their genetic material inside the DNA of living organisms. You can imagine viruses as tiny but powerful players in Earth's biological system, quietly influencing processes that affect the entire planet. Scientists continue to discover new viruses and learn more about how they function. Each discovery reveals that the viral world is far more complex and widespread than previously imagined. Despite their simplicity, viruses remain one of the most fascinating and influential forces in nature.